Thank you, Geraint, for inviting me to this webinar. Thank you, Goetz, for a very kind introduction. There was a lot of things I think I don't deserve that much credit for that, but it is a very hot topic nowadays, which is going on in the European Union about the regulation of NGT, new genomic techniques in plants, and I'm happy that I can talk about it, give you a lot of new information, but unfortunately, I cannot give you the final answer what's going to happen. Regarding my work, I recently addressed the topic under the name of European Federation of Biotechnology, where I was leading a special focus group on that task, and also in the framework of a new project, Tangents towards next generation crops, which was recently awarded to us from operational programs under the scheme of Johannes Amos Comenius, famous teacher in the Middle Ages. There are several work packages, and I'm leading the work package outreach, which is actually devoted to spreading more information, knowledge among public, and educating people in order to understand new technologies. So it is actually my job to talk about and try to explain these things to people. And I was happy to be invited also to European Parliament to trying to explain some things also to MEPs in the Parliament. Next slide. I have some problems with changing the slides then. Okay, this way. Sorry. Okay, so I'd like to start with, you all know what NGTs are, but to look forward to the possible use and importance, it's not so easy because they actually were not being put into practice yet. And if we can look at the comparison or to what they can do and can project what they can do, it's possible to compare with conventional and GMOs and other techniques we know and we were used for plant breeding. So I borrowed this figure from one recent review and also put some my own notes into it. And if you look at what people are doing, have been doing for 10,000 years to create the crops, they were using crossbreeding all the time. With all the new knowledge and technologies accumulated in the end of the 20th century, it was actually possible to see diverse crossable species. Then there were already varieties that were created, they were very well selected, and it was possible to transfer various traits of interest into the allied variety by crossing. But with this, of course, it goes according to the laws of genetics. So it takes a lot of time and a lot of cycles of crossing to obtain what you want. And very often, you don't get only transfer of that particular gene or genetic information, but you get a lot of other information you cannot actually remove by crossing, and sometimes can be unwanted and can bring traits which are not good. So extensive selection and a lot of field test and a lot of testing and selection is needed. It takes a lot of time, usually up to 10 years, and a lot of effort. And actually, already the possibilities are limited, but in many ways. Then during 1950s, there was another technique that developed, was used for increasing diversity of the population. It was used for breeding by rather drastic methods, either chemical or radiation mutagenesis. So people created a lot of mutants, and those which survived were tested for selected traits, and from those, again, the new variety was selected. Again, it took a lot of time. There were, of course, necessary many trials, but nowadays many crops are actually based on these mutants. Then in 1990s, GMO plants hit the market, and they are based on genetic transformation, usually transferring foreign gene, mostly from bacteria, to the plant. The technique is actually bringing or integrating a new gene or piece of genetic information randomly in the plant genome. Usually it's only on one chromosome, but there could be multiple copies. So typically you get a heterozygous plants where which you have to reproduce, you have to select for homozygotes, and again, it takes you a lot of time to stabilize the line you want to obtain. So again, it takes about 10 years and a lot of effort to get what you want. On contrast, NGTs are based on a technique using CRISPR-Cas, which makes breaks in DNA. Breaks are repaired randomly. Then you don't get so many mutants because this is targeted, not random, as the all other techniques. You don't get too many different variants. In our experiments, for example, with barley, we usually get about 10 to 20 different outcomes. So it's much easier to select. And also important thing is that if the construct is prepared properly or well targeted, it makes actually it hits all the genes of that sequence, all the DNA of that sequence. So you can get homozygous plants very quickly. So it actually decreases labor and decreases the cost of the breeding. And very importantly, it can also shorten the breeding cycle to about 1/2. So the advantage is clear. You get targeted mutagenesis, you mutate what you want, you can bring the trait what you want. Then again, you are much faster, it's much cheaper. And one more important thing is that you can get advantage in the race against pathogen evolution. Because every time pathogens can overcome resistance if you breed new varieties, but if it takes you 10 years, sometimes the pathogen can overcome it in 5 years or 3 years. So you should be as fast as possible, of course, with lowest or minimum expenses. So that's why NGT is so widely discussed and so widely developed now. Then this was a introductory slide, actually, which is actually the longest thing to talk about. And now the facts. EUSAGE is a network which is collecting all the information about research results on NGT plants. And you can see in the database, they already recorded that, okay, rice has about 160 different mutations, tomato, maize, soybean. So there is a database of plants that can be mutated that way, and that can get new traits. And these new traits could be improved food or feed quality, yield and growth, stress tolerance, and many other quite positive, interesting traits. So you can see that this is, of course, very interesting for plant breeders, and it's could become a new tool. And more importantly, most of these new traits are based on very simple modification of the genome. So if you look throughout the database, you found that with genome editing, you will get over 99% of small modifications like SDN1 and SDN2 changes. So very, very small modifications. And these modifications can actually, or are actually appearing in the genome elsewhere by natural evolution. In contrast, longer DNA sequences, if they are introduced, they usually resemble GMOs or introduction of inserts which are typical for GMO plants. Then if you look at the map, how these techniques are regulated, you can see the green countries where they already have the legislation. You can see the countries which are considering or discussing the legislation. You can see Europe in red, which has the legislation based on GMOs. So quite old legislation. Then how this all started? I mean, we all were very much surprised that in 2018, European Court of Justice actually ruled that all new NGTs or plants created by NGTs should fall under the category of GMO. They should be regulated as GMO. It was, for scientists, it was a surprise because regulating the new technology invented in 2010 and later on, according to the law from 2001, is a little bit strange, but for lawmakers, it makes perfect sense. But many scientists started to protest and started to write papers and letters and made petitions. So finally, European Commission started to move, and this started a study on new genomic techniques in February 2020. Then they invited stakeholders, various international associations, interest groups, and learned societies to give expert opinion and questionnaire. There were actually 54 of such stakeholders, and EFB, European Federation of Biotechnology, is one of them. So that's why we got into the business. It took 1 year. It was a COVID year, so it was quite long. But finally, in April 2021, European Commission published the results of the study with saying that the legislation is indeed old and should be updated. And the new techniques are as safe as random mutagenesis. So they are perfectly safe for creating new crops. This was applied only to plants, not bacteria or animals. And also the European Commission noted that actually new technologies are needed to fulfill the goals of the Green Deal, bioeconomy, sustainable agriculture, etc. So at the same time, they started a legal initiative for certain genome-edited crops. And that legal initiative involved also public consultation, and again, consultation with various stakeholders, including EFB. So we're again trying to give our best opinion, filling questionnaire. We were invited for interview. We were asked how we project use of these technologies for 2030, how we can imagine what will be happening in agriculture sector in 2030. We did our best. It's not so easy, of course, to project the future, and to that extent. And finally, the Commission sent a legal proposal, so amendment to the present law, to European Parliament in July last year. Then Interparliament, it went immediately to discussions, and the proposal was adopted by Agriculture Committee in December last year, then by Environmental Committee in January this year. You can always see that how many votes there were in favor, against, and abstained there. So the majority was overwhelming in Agri Committee. It was not so big, but still good in Environmental Committee. And finally, the plenary vote took place on February the 7th. And the proposal was adopted by narrow margin, actually. During the process, there were so many amendments, changing a lot of things, but in general, these amendments were quite positive and improving the original proposal of the European Commission. Now, then Okay, then the adopted proposal was sent to European Council, and there they are debating, discussing what to do about this proposal, and they need to reach a consensus. So last week I was following, 2 weeks ago, I was following the debate of the council. There were interesting views, and they are starting so-called informal trialogue between the Commission, Parliament, and the Council to get the final proposal that would be submitted to the European Parliament for the second reading. Due to the time and coming elections, I'm afraid that it will not be possible to do it in April 2024, which is actually now, and the session of the Parliament is, I think, in the last week of April. So, what is the content then? Most important thing is that the proposal introduces two categories of plants, NGT1 and NGT2. NGT2 is almost equal to GMO. So there's not much new how this would be regulated. But NGT1 is very much, I would say, liberal compared to NGT2. And so into the category, it is important how many mutation events are allowed. And it means that up to 3 mutations per any protein coding sequence are allowed. And these mutations can be substitution, insertion of no more than 20 nucleotides, or deletion of any number, which is quite a lot. And we were trying to explain the members of the Parliament, this is actually happening in nature. Actually, in nature, you can get a lot of mutations of on small scale, and still this proposal is still restrictive to what is happening in the nature, because there are, of course, many other possibilities. But let's make it simple. So this is something which is the core of the proposal, and it's in Annex 1. Then important thing also in this annex is that DNA sequence which exists in a gene pool of breeders of that plant or crossover plant can be actually freely relocated in the genome. So it can be inserted, it can be substituted, it can be translocated or inversed. And this is also something which is happening during the breeding process. So that's fine here. NGT1 then should be treated like a conventional plants. Important thing is that many Parliament members don't want these new varieties to be patented, but unfortunately, the amendment in the plenary vote also introduced compulsory labeling of those plants. Then situation in Council. During the Spanish presidency of the Council, we're actually testing the position, and in order to get the law adopted in the Council, you need at least 15 countries in favor, and those countries should have 65% of the population, which is so-called qualified majority. Then, in December, there were position of 18 countries which are in favor, 7 against, and 2 abstain. These 2 abstained was Germany and Bulgaria. And then, unfortunately, the population quorum was not reached because it was only 60.55. Then, at the meeting of the Agri and Fisheries Council in March, most of the countries actually repeated their standpoint from December. Presentation of the Germany was very short, that they welcome the proposal, but there are still questions. From Poland, they were saying, okay, they can, it's necessary to change some things, and but it was not clear if they would be willing to change their vote. So it's still under discussion, and we need actually Poland or Germany to overturn the decision at the moment. So this is the end of my talk. We are waiting, of course, what's going to happen in the European Council and Parliament. And I thank you for your attention. I'd like to also remind you that European Federation of Biotechnology is running European Congress on Biotechnology, which will take place in beginning of July in Rotterdam, and we are all invited. Thank you very much.
Okay, thank you, Ivo, for this nice overview. So I will see in the chat if there are questions. I see one question of Tobias Broekman, which was already more or less answered by Geraint, about the EU-like regulations that still apply to Scotland and Wales, and that deregulation is only being discussed for England. This is often unclear on maps. Can you confirm this, or do you have other information? I can read what Geraint answered there. So he said that the question is indeed rather specific, and he doesn't know if you might know the answer. But yeah, sorry.
I'm sorry, I don't know. I know that the regulation is already valid in England or UK. I don't know how it applies to Scotland or Wales. I'm sorry, I'm following the European Union. So
I can give some input on that. So I was recently at a meeting in Scotland, and someone from Scottish government there mentioned that they, even though at the moment their laws will stay the same as they are, so they will ban GE, but they will take their lead from the EU. So if the EU changes, then Scotland will change as well, and I suspect that will be the same for Wales as well.
Yeah. Then there is a question of Malcolm Burns. If you have a plant produced by NGTs, which consists of three mutation events, each of 20 nucleotides, would it still be classified as an NGT category 1 plant?
Yes, that's correct. There could be 3 such events per protein coding sequence, actually. So not only for the whole genome.
Okay. I don't. And what kind of labeling is required?
Well, this is still widely discussed. The original proposal that was that labeling of seeds, which are distributed to farmers and to users, should be labeled, so kind of labels on the seed bag, and also registration will be required. But some interest groups want to change it to the labeling of products. At the moment, the amendment is not that clear, and I still think that it's valid only for the seeds. But this will be probably the topic of the debate in the council.
Okay. Then there is another question. Have NGTplants been investigated again? Plant pathogens, against pathogens, I guess. How effective are these NGTplants against biotic stress?
Yeah, I can already read the chat. This is very general question, of course. There are examples where a single gene can be mutated in a way that the whole variety becomes pathogen tolerant. So there are examples like this.
Can I ask a question, Katrijn?
Yes, of course.
Yeah. So can I just ask for a bit of clarification about what you said about a German example? So and the requirement to have a certain number of countries versus a certain number of populations. So that means, for voting purposes, the countries with the largest populations have the greatest influence on the vote, I would assume. So therefore Germany is quite key, given it's the largest population in Europe. Is that right?
Yeah, it's right. It's so-called qualified majority in the council. So it's decided you need to have at least 15 countries in favor and 65% of population at the same time.
Mhm.
So we need actually 4.5% of population at the moment to get this through. And we already have over 60% of population in favor in a European Union, which is, I think, quite good.
65% of the population are not reached in the final evaluation.
Well, the amendment will not be adopted. But this is not like voting in a parliament. This negotiation can take time until the consent is reached. So they can meet many times until the consent is reached, and then the final adopted proposal will be sent back to European Parliament for plenary vote. Unfortunately, we can get the delay because the next presidency is Hungary, which is strongly against NGTs. And of course, the elections can change things in the European Parliament.
Okay, there was also a question before of Molly Langdon about the regulatory sequences. So they are excluded from these limits in the annex. Does it mean that editing and promoters will not be regulated?
Well, that's very good questions. When they were preparing the definition, they were trying to include everything and limit the changes by different means. So now it's very clear that the changes in the protein coding sequence only count. So you are probably right that modification in the promoter region would not be taken as a modification. This is something that maybe need to be clarified. And I see a similar question in the chat. How is classified a mutation of the protein coding region? Same mutation of 2 nucleotides besides one of 30. If there is a mutation of 30 nucleotides, it's NGT2.
Okay.
Of course, unless this is this sequence comes from breeders' pool.
No. Okay. I don't see any more question in the chat. So I guess, yes, there is one. Maybe we take that one and then when during the other lecture there comes some other questions, we can answer them in the chat. So why not all countries from Europe were participating in the voting? 1 year ago in Ukraine was approved new law about gene editing and GMO plants.
Because this is a regulation of the European Union, so not Europe. Not the whole Europe. European Union, Europe is not the same.
Okay. Voilà. So thank you, Ivo, for your lecture and answering the question. And now I give the floor back to Goetz to introduce Bingyang and then listen to Bingyang lectures. Thank you.